All right. Good morning once again. So we proceed with our test one physiology essential questions on essay and short notes. So next we are going to look at three questions under nerve physiology. Three questions under nerve physiology. So the first question that we're going to talk about or look at is on question number 16. Okay, is on question number 16. As far as nerve physiology is concerned. Okay, so question number 16 says describe the three functional types. Okay, take note of this word functional types. Says describe the three functional types of neurons using relevant examples. So this is nerve physiology. Okay, so anyone to give us the three functional types of neurons? Three. Three functional types of neurons. Anybody? Anyone to give us the three? Okay. Sialuya and Muchimba. So they've given us sensory, motor, intermediate. So all of them, they've given us the same answers. Thank you, Muchimba and Sialuya. So basically, those are the uh, we've also received someone who has used just different terms, more like instead of saying the same word, you just use synonyms. So someone else has said afferent, efferent, and intermediate. Okay, so those are the three functional types of neurons that they want you to describe in this particular question. Okay, so neurons can be classified into three functional types based on their role when it comes to transmitting information. But remember, when it comes to classification of neurons, is that you can classify neurons based or depending on the number of pores that they have, right? That is where you have unipolar, you have bipolar, and you have multipolar. Then they can also be classified depending on the length of their axon. That is why you have the Golgi type 1 neurons and you have the Golgi type 2 neurons. Okay. But now this question is focusing on the classification that depends upon the function. So classification based on function, we have what we call sensory neurons. So sensory neurons are also known as afferent neurons. Okay. So now, what is the function of sensory neurons? So the sensory neurons actually, they just transmit sensory information from the sensory receptors. So they take that information from the sensory receptors to the central nervous system. Now we've been asked to give um, relevant examples. We have the photoreceptors. Okay, we have the photoreceptors. So these photoreceptors, they are found in the retina. Okay, the retina has to do with the eye. Okay, so they are found in the retina, and what they do is that they transmit visual information to the optic nerve. I know you might not relate as we are talking about the optic nerve, but for now, just know that the optic nerve, okay, just know that we're talking about the optic nerve. So to the optic nerve, then we also have auditory neurons. So auditory neurons have to do with transmission of sound information from the cochlea to the auditory nerve. Okay, then we also have the nociceptors. Okay, so these ones they transmit pain from the skin to the spinal cord. Okay, that is on sensory. Then we also have motor neurons. So motor neurons are also known as efferent neurons. What is their function? So their main function is just to transmit motor signals. So for motor neurons, usually these are concerned. Motor is concerned with the movement. Okay, motor is concerned with the movement, locomotion. So what happens is that the motor neurons they transmit signals from the central nervous system now to the muscles or to the glands. So when we say efferent, from the word effector, okay? So what happens is that these muscles or glands are what we call effector organs, okay? So examples include we have the alpha motor neurons. So the alpha motor neurons they tend to transmit signals from the spinal cord towards or to the skeletal muscles. We have beta motor neurons. We transmit signals to spinal to muscle spindles rather, and we also have uh, autonomic motor neurons which transmit signals to smooth muscles and the glands. Then lastly but not the least, we have interneurons. So interneurons are also known as the intermediary neurons or intermediate neurons as most of you had put it. Okay, so their function is just to process and integrate information within central nervous system. Examples include the cerebellar interneurons, which integrate sensory and motor information for coordination. We also have spinal cord interneurons, which process reflex information, and the cortical interneurons, which integrate sensory information for perception. We are done with that question. Are we good to go? Can we look at the another question? Okay, so the question was about describe the three functional types of neurons using relevant examples. Describe the three functional types. Okay. Okay, so someone is asking, what about the issue of Enlager-Gasser and Lloyd-Hunt classification? Where do they fall? I think I'm going to that question. I think I will tackle it. If I won't talk about it, if I don't have it in this file, then I will talk about it. Is that okay? All right. So we jump to question number 35. Remember, we're talking about nerve physiology here. Okay. So we go to question number 35. So question number 35 says, describe, okay? Describe and distinguish between unmyelinated, these are the key words, unmyelinated and myelinated neurons. Unmyelinated and myelinated neurons, and the differences in the ways through which they do what they conduct impulses. So these are basic informations that you need to know with regards to as long as you've done nerve physiology. Okay. So now neurons can either be myelinated, which is true. Okay, they can either be myelinated, just as we have seen from the question, or they can be unmyelinated. Okay. Now, when we are talking about the unmyelinated neurons, okay, let's talk about the unmyelinated neurons. The unmyelinated neurons, what happens is that they lack myelin sheath. Okay. So what we're trying to say is that these unmyelinated neurons, they do not have a myelin sheath that surrounds their axons. Also, unmyelinated neurons, they tend to have a smaller diameter. So what that simply means is that unmyelinated axons are typically smaller in diameter. Then when it comes to their to the rate of conduction, the speed, the velocity, is that the unmyelinated neurons actually conduct impulses um, at a slower rate. Remember, myelin sheath around axons helps improve the rate of the impulse conduction. Okay, then we've been told also to talk about the way through which conduction occurs. So in unmyelinated neurons, the conduction occurs through what we call continuous conduction. Okay, so you'll find that action potentials are generated continuously along the length of the axon, and this also actually contributes to it is one of the explanations why the conduction velocity is slower in unmyelinated neurons, because what will happen is that just this rough illustration. So you find that since this axon, okay, this axon doesn't have myelin sheath, what will happen is that the impulse will have to move all the way, finish all this and to reach where it is going. Okay. But you see when we talk about myelinated neurons. Then when it comes to ion channels, the voltage gated ion channels are distributed uniformly along the what? The axon. Then let's talk about myelinated neurons. So myelinated neurons, they have myelin sheath surrounding their axons, and the myelin sheath is contained of lipids and proteins. Okay, and them, in contrast to unmyelinated neurons, is that they have a larger diameter. Okay, then when it comes to conduction in myelinated neurons is that it is very faster. Okay, we have faster conduction velocity. Remember, myelin sheath improves the velocity of impulse conduction. Then, in contrast, remember, under unmyelinated neurons, we said that the mode or the way through which they conduct the impulse is through continuous conduction. But when it comes to myelinated, we have what we call saltatory conduction. Okay, we have saltatory conduction. Why? Because you find that the action potential will tend to jump from one node of Rambia to another, allowing faster transmission. Okay, so you'll find that let me take you back where I had. Okay, so here you'll find that if we have myelin, okay, we have myelin sheath here, we have a myelin sheath. Okay, so you find that instead of moving through all this, if the impulse is here, it will jump and come here. Are you seeing that? So from here, instead of moving, it will jump like that. So as it jumps, you'll find that it moves even faster than in continuous conduction. And then in myelinated action neurons, rather, the voltage gated channels are concentrated at nodes of Rambia. Okay. So when it comes to key differences, you'll find that in to key differences, you'll find that conduction velocity differs. Okay, energy efficiency, myelinated, they'll require less energy for impulse conduction than axon diameter. Myelinated axons are larger in diameter. Presence of myelin, we've already established that fact. Can we proceed to the next question? Okay, so now we go to question number 40. Let's jump to question number 40. Okay, we jump to question number 40. Muchimba, yes. So Muchimba, this is the question that you were talking about. Okay, so with regards to this question is asking you, saying that, so this is a rare question, but if it finds you, most people fall victim because it is rarely asked, but it's something that you need to know. Okay, under nerve physiology. So the question says, discuss the classification of nerve fiber types. Discuss the classification of nerve fiber types. Okay. So have you seen? So this one specifically tells you to talk about the classification of not neurons, but it talks to you about the nerve fiber types. Okay. So you'll find that mammalian nerve fibers are actually categorized into or classified into A, B, and C groups. Okay. Now, further, the group A is divided into alpha, beta, gamma, and delta fibers. Okay. Now, in general, the greater the diameter, okay. Remember, we've already established here. Oh, it's very far. The question is very far. Remember, we've already established that which one was faster between myelinated and unmyelinated neuron? Which one was faster between the two? Which one was faster between the two? Between unmyelinated and myelinated axon. We said myelinated, right? And which one had a larger diameter between myelinated neuron and unmyelinated neuron? Myelinated. Okay, so what we're trying to say is that the greater the diameter of a given nerve fiber, the greater is its speed of conduction. Okay, and you must understand one thing, that the, the large axons are usually concerned primarily with what we call proprioceptive sensation. Are we together? Are people following? Yes. So they are concerned with proprioceptive sensation, somatic motor function, conscious touch, and pressure. But when we're talking about those which have smaller axons, those ones they subserve pain and temperature sensations and the autonomic function. So when you are told to discuss or to talk about the classification of nerve fiber types, everything is summarized in this particular table, okay, with regards to the types of mammalian nerve fiber types. So we have A alpha. So A alpha, its function is actually concerned with the proprioception. Proprioception, okay. Hopefully we are together. So when you're talking about proprioception, it just has to do with how can we put it? It has to do with your body's ability to actually sense movement, action, and location. Okay. So in other words, proprioception in physiology has to do with the perception or awareness when it comes to yes, proprioception has to do with the perception or awareness of the position and the movement of the body. Okay, are we together, Mchimba, on that? So you'll find that there are times when you've not seen that someone is coming, but you are able to tell that, oh, there is somebody behind me. Okay, that is proprioception. Then we also have somatic motor. Okay, we also have somatic motor. Then when you look at the diameter of the A alpha is 20 or 12 to 20 micrometer. Then the conduction velocity, I'm sure you can see that it is the highest here. Okay, it is the highest. So you can talk about the fiber type and its function. Then you also describe its diameter and also the velocity conduction. Then alpha beta is for touch and the pressure. Then its velocity like that, um, uh, and also the diameter. Then you also have the alpha gamma, which has to do with motor to muscle spindles. That is the diameter 3 to 6. then the speed is 15 to 30 uh, meter per second. Then you have the A delta. So the A delta has to do with pain and temperature. Okay. Then that is the diameter 2 to 5 micrometer. Then the speed is 12 to 30. Then you have the group B. So group B has to do with preganglionic. So you also have preganglionic. Okay, you have a preganglionic uh, automatic. Hopefully, we know what that means. Okay, so when you're talking about the preganglionic neurons, actually, these are the first of two neurons in the automatic nervous system. Okay, and them they actually come from the spinal cord and the brainstem. Okay, now these preganglionic neurons, or you must understand that they are usually cholinergic. Okay, they are usually cholinergic, and they usually use acetylcholine as their neurotransmitter, and they are typically myelinated. Are we together up to this far? Are we together? Are we following? Okay, so there's a question. There's a question I think that I'm seeing in the. Let me open the comment section. Someone has just commented. Okay, someone is saying, is C fiber also known or called type 4 fiber? And why is it not myelinated? Why is it not myelinated? Anyone? Any takers? So first, let's tackle the first part, which says, is the C fiber also known as the type 4 fiber? Anyone respond to the first part because it is very straightforward? Anyone to answer that and give us the reason? Okay, Muchimba is saying yes. Emmanuel, have you seen the response to that? Okay. And then Muchimba, can you also now tell us why are they not myelinated? Is there anyone who can tell us? Okay, so the the answer has already been given to the first part of the question, and the reason why they're not myelinated is not function doesn't require rapid signal um, transmission. Okay, I like the line of thought. Okay, I like the line of thought. So I think what the line of thought of for Muchimba is okay. Okay, so we've said that these are also known as type C fibers. Okay, and the reason why they're not myelinated, the first function, or it is the first reason, is what Muchimba has said when it comes to slower conduction, because These ones, they actually transmit signals more slowly. And remember that when um, an fiber has been myelinated, it means it will conduct at a very speed rate. Emmanuel, is that okay? Is Emmanuel still in the meeting? Is that okay? Yeah, so they tend to transmit signals at a very slow rate, which is very cardinal or very appropriate when it comes to the type of information they carry. Okay, for instance, you know, dial information, temperature, and the like. Okay, so type C, we have the dorsal root and the sympathetic. I think we are done with that question. I think we are done with nerve physiology questions that you might expect. Are we good to dive into something else as well before time runs out? Okay, so we are going to. So, by the way, under nerve physiology, some of the things that you should be able or be conversant with has to do with glial cells. Okay, the glial cells know their functions, uh, the oligodendrocytes, the Schwann cells, the astrocytes, all those glial cells. Okay, know them and know their functions and where they are found. Know the classification of neurons, very, very important. Okay, hopefully we are aware of that. Then another thing that you should understand or take note is that when we have what we call hypokalemia and we have hyperkalemia, know those things as well. Very, very important. Okay. Then another thing also, we have the physioclinical classification, okay, of the nerve fibers. Okay, this is not there in my notes, but I'm just telling you that you need to know the physioclinical classification, which has to do with susceptibility of these fibers, because you have uh, type A, B, and C. Okay, so you'll find that when it comes to hypoxia, type B is the most susceptible, and then type C is the least susceptible when it comes to hypoxia. Okay, when it comes to pressure, type A is the one that is the most susceptible to pressure. Then the least is type C. Then when we're talking about local anesthesia, the most susceptible is C, and then the least susceptible is type A. Very, very important to take note, thought of highlighting those things. Are we together? All right, so we move on to question number 36. By the way, we are done revising nerve physiology. I want to talk about the action potentials and all those, but you need to know them. I think there is a video, a recorded lecture that is on our YouTube channel with regards to all the details of action potential in neurons. You can check it out if at all you missed it. Okay, so one of the peculiar aspects when it comes to synaptic and junctional transmission, one of the things that you need to know has to do with intercellular connections. Okay, to do with intercellular connections. So this question is asking us to describe. Someone is is asking about the neuro the neuromodulator. Is the neuromodulator important to know? Okay, so when we're talking about neuromodulators, by the way, if someone wants to know about that, these are just substances which are very important when it comes to if we can modulating or altering or taking part in controlling the activity of neurons. Okay, so you'll find that they tend to influence and they when it comes to transmitting nerve impulses, but actually they do not initiate the transmission. Okay, so when it comes to neuromodulators, you can just know some of them, which what they do, but yeah, you can just know some of them and what they do because you have one of the most common neuromodulators. You have, I think, you have acetylcholine, you have serotonin, you have dopamine, you have no adrenaline. Okay, those you can just know them and what they do. Okay, for instance, acetylcholine is very important. You already know that it is very significant when it comes to muscle movement. Okay, can we now proceed? Emmanuel, can we proceed? All right. So describe the intercellular connections. Describe the intercellular connections. So intercellular connections are also known as cell junctions. Okay, are also known as cell junctions. So now these cell junctions, I believe you understand and you learned that they are specialized structures which tend to link adjacent cells. And when they link adjacent cells, what they do is that they bring about or they enable communication and also exchange of molecules, including mechanical support. So we have various types of intercellular connections. We have various types of intercellular connections, some of which include we have tight junctions. Okay, so tight junctions. You need to one of the things that I want you to understand or remember is that they seal adjacent cells. So when they seal adjacent cells, they prevent free diffusion of molecules. Okay, so the main function of tight junctions is to maintain epithelial barrier. Okay, regulate permeability. Where are they located? In epithelial cells. Then the other one that we have has to do with adherence junctions. Okay, so these ones they mediate cell to cell adhesion. Okay, what is the major function? Is to maintain tissue structure and also regulate cell migration. Where are they found? Epithelial, endoepithelial, and muscle cells. Okay, then when you talk about desmosomes, so desmosomes, these are strong adhesion complexes. Their function is they help in resisting mechanical stress and also they maintain tissue integrity. Where they found epithelial tissue, cardiac, and skeletal muscle cells. Then we have gap junctions, which most students don't forget. So gap junctions are the ones which allow direct communication between cells. Function, there is the exchange of ions, okay, metabolites, as well as the signaling molecules. Okay. Then you also have hemidesmosomes. So hemidesmosomes, they anchor cells to the basement membrane. Okay. They are found in epithelial cells. So these are key molecules that are found in these intercellular connections that we've talked about. So we have claudins. Okay, so claudins, you will find the molecule known as, I mean, in tight junctions, you'll find the molecule known as claudins. Then in AJ, you find molecules known as cadherins. Then in desmosomes, you have desmogleins. Then in gap junctions, this one most people don't even forget about it. In gap junctions, we have connexins. Then when it comes to hemidesmosomes, the key molecules are integrins. Are we together? Can we proceed to the next question? Can we proceed to the next question? All right. So the next question that we are going to go to is question 41. Okay, question 41. Okay, so this question 41, I just made it deliberately. It is not how this is not how it can be phrased in your assessment or in the exam. But if I was your lecturer, I would phrase it the way it is, like this. But I put it so that you can get to understand the various steps when it comes to the neuromuscular junction. Okay, are we okay? Are we together? Are we together? Yeah. So the question says discuss the physiology of the neuromuscular junction under the following headings. The first one is action potential propagation. The second one is um, acetylcholine release. The third one is generation of end plate potential. The fourth one is action potential propagation in muscle fiber. The fifth one is muscle contraction. Then the sixth one is termination of signal. So when you're talking about muscle contraction, this can be properly exhausted when we are talking about muscle physiology. But on this particular question, we'll only tackle it a little bit. Okay, are we together? Can we now can we proceed to answer the same question? All right, cool. Okay, so when we are talking about the neuromuscular junction, this is actually just a specialized synapse where we have a motor neuron communicating with a muscle fiber. And when they communicate, what happens is that um, it will bring about muscle contraction. Okay, so when we're talking about the physiology of the neuromuscular junction, which is NMJ, it involves a wide range of a series of events starting with the action potential, traveling down the motor neuron, reaching the synaptic terminals. Now, let's focus on what the question is asking us. Okay, so action potential generation. So, under action potential generation, what we can say is that the action potential travels down the motor neuron axon until it reaches what? The nerve terminal. Okay, and when it reaches the nerve terminal, you'll find that the action potential will depolarize. By this time you understand what these terms mean. Will depolarize the nerve terminal, and when it depolarizes the nerve terminal, it will open the voltage gated calcium channels. Okay, now when the voltage gated calcium channels open, you'll find that calcium ions will eventually enter the nerve terminal. And when it enters the nerve terminal, what it will do is that it is going to trigger the release of neurotransmitters. Okay, so in other words, the release of neurotransmitters into um, the synaptic cleft or that space is dependent on calcium ions. Very important. Okay, so in other words, to just reiterate on what I've said with regards to action potential propagation, is that when the action potential reaches the synaptic terminal, the voltage gated calcium channels will open, and when they open, they will allow calcium ions to flow into the presynaptic cell. Okay, then you find that the increase in intracellular calcium ions would trigger the release of the neurotransmitter, which is acetylcholine, from the vesicles into the synaptic cleft through the process known as exocytosis. So, with that being said, it leads us to the second subheading, which is acetylcholine release. Okay, so you need to understand that acetylcholine actually is synthesized from acetyl-CoA and choline in the nerve terminal. Okay, and when it has been synthesized, it will be stored in synaptic vesicles. Okay, now after calcium has influxed or into the cell, it will trigger the release of acetylcholine. So that release is into the synaptic cleft. Okay, so you'll find that acetylcholine actually is released in discrete packets, which we call quanta, corresponding to the single synaptic vesicles. So to reiterate, what I have said is that when it comes to acetylcholine release, you need to know that acetylcholine diffuses across the synaptic cleft. And when it diffuses across the synaptic cleft, what will happen is that it will bind to what we call nicotinic acetylcholine receptors on the muscle fibers membrane. Okay, so you'll find that it is these receptors are actually the ligand gated ion channels that when acetylcholine binds to them, they'll allow the influx of sodium and the efflux of potassium. And when that occurs, you'll find that it will lead to depolarization of the muscle fiber membrane. Okay, then that will lead us to our third subheading, which is generation of end plate potential. Okay, so you'll find that I've already mentioned that the released acetylcholine will bind to what we call nicotinic acetylcholine receptors. So those nicotinic acetylcholine receptors are found on the muscle fiber surface. Okay, and then the activation of the nicotinic acetylcholine receptors will open the ligand gated ion channels. And as such, sodium ions and calcium ions enter the muscle fiber, thereby depolarizing it. Now, this depolarization that will take place will generate what we call end plate potential. So, the end plate potential is just a localized potential change. Okay, so the depolarization that will occur to bring about the end plate potential is due to the influx of sodium ions through the nicotinic acetylcholine receptors. Okay, now from there we go now to the other subheading, which is action potential propagation in muscle fiber. Okay, so what happens is that if the end plate potential gets to reach threshold, an action potential will actually be what? Be generated. So after it has been generated, it will need to be propagated in the muscle fiber. Okay, so what will happen is that the action potential will spread along the muscle fiber. Okay, so this is actually driven by the voltage gated sodium channels. Okay, and what will happen is that this action potential will actually or eventually depolarize the entire muscle fiber. Okay, so in other words, the EPP triggers the voltage gated sodium channels along the muscle fiber membrane. And when that happens, it initiates an action potential that will propagate along the muscle cell. So what will happen is that this action potential that has been generated will travel along the T tubules. The T tubules are found in the muscle cell, and when that happens, they will lead to the release of calcium ions from the sarcoplasmic reticulum into the cytoplasm. But I don't want to dwell much to go deep into that. I think we'll talk about it when we are looking at questions under muscle physiology. Okay. Then after that has occurred, it will lead to what we call muscle contraction. Okay. So with regards to muscle contraction, you need to understand that the action potential that has been generated will trigger the release of calcium from the sarcoplasmic reticulum, just like I earlier mentioned. Okay, so for us to have muscle contraction, calcium ions will actually bind to what we call troponin. Okay, will bind calcium binds to troponin, and then we have tropomyosin there, whereby it is going now to expose myosin binding sites. Okay, now for us to have contraction as well, actin and myosin get to interact, and when they interact, they produce muscle contraction, which will be talked about in detail under the sliding filament theory. Okay, so in other words, the rise that we had, or that we're going to have in intracellular calcium ion concentration, would trigger the interaction between actin and myosin filaments within the muscle fiber, and when that occurs, it will lead to muscle contraction. Okay, so you'll find that this process of muscle contraction actually it involves the binding and the hydrolysis of ATP, and ultimately the shortening of the sarcomeres, resulting in muscle contraction. Then lastly, we have termination of the signal that brought about the contraction. So if that signal is not terminated, the muscle will be in perpetual contraction, but we don't want that to occur. So what will happen for us to, for the signal to be terminated? There's what we call acetylcholine esterase. So acetylcholine esterase will break down acetylcholine into choline and acetate. Okay, so when that has occurred, you'll find that the nicotinic acetylcholine receptors will become desensitized, okay, whereby now it will reduce further signaling. And when that has occurred, remember calcium had gone to troponin. So what will happen is that the calcium will be taken back to the sarcoplasmic reticulum through what we call sequestration. So in other words, the calcium will be sequestrated and muscle contraction will cease. The muscle will relax. Is that okay? We are done with that question. Are we good to go to the next question? So we've summarized action potential up to muscle contraction. People just want to know the question. Okay, so the question was discuss the physiology of neuromuscular junction. Okay, so um, I believe though that was the last question on synaptic and junctional transmission. Now I think we're going to look at last two questions, and they'll come from muscle physiology. Okay, can we proceed to the next question? All right. So the next question is question number 10. The next question is on question number 10. So question number 10 talks about, is saying, explain the process of skeletal muscle contraction in humans. Explain the process of skeletal muscle contraction in humans. So, as I've already earlier alluded to you, that the way I'm bringing out the points is is to give you a clear picture of what you are supposed to write, but that means that you should also be putting headings. Is that clear? But it's just to give you a proper guidance. Is that okay? Okay, great. So, in step one, according to, firstly, I would want to mention that when it comes to muscle physiology, um, there are a few things that you need to know. Okay, you need to know a few things. Okay, you need to know the differences in the three types of muscles. Okay, you need to know the mechanism of muscle contraction in skeletal and in smoothie muscles. That you need to know. Okay, and also, yeah, I think that is very, very important for you to know. And when it comes to the structure of the skeletal muscle, you need to know about the structure and the function of the skeletal muscle. um, you need to know about the structure of what we call the sarcomere. Okay, you need to know about the sarcomere, very, very important. Okay, that you need to know. The sarcomere you must know, very, very important. You must know more about neuromuscular transmission. That is why you have the neuromuscular junction. Okay, you must know about what we call the excitation contraction coupling. You must know that. Okay, very important to actually know. Okay, then you also need to know the sliding filament theory. Also know the clinical correlates, where you have things like myasthenia gravis, you have Lambert-Eaton syndrome. Okay, need to know. You also need to know things to do with muscle mechanics, where you have twitch, summation, and tetanus. Those are part of muscle physiology. Very important. Also need to know about muscle fiber types. Okay, but nevertheless, that is not where the question that we have to answer is focusing. I just wanting to let you know about some of the things that you must know in muscle physiology. Okay, explain the process of skeletal muscle contraction in humans. So my first heading here is talking about neural stimulation. Okay, so the the process actually begins with the transmission of a neural signal from the central nervous system to the muscle fiber. Okay, then the neural signal or the action potential travels down the motor neuron and reaches the neuromuscular junction. Okay, I think we've explained that in the previous question, so we already know how uh, the action the action potential travels and the like. So you connect that same knowledge. Are we together? Are we together on that? Then step two is concerned with the neuromuscular transmission. Neuromuscular transmission. So what happens under neuromuscular transmission is that we have what we call neuromuscular transmission. It talks about at the neuromuscular junction. So I think let me dwell. Can I dwell a bit on the neuromuscular junction so that you can have a proper and clear orientation with regards to the neuromuscular junction? Can I dwell a bit on this? Okay, so when we are talking about the neuromuscular junction, as far as muscle physiology is concerned, you need to know that the alpha motor neuron is the one that supplies the muscle fibers. So what happens is that when the action potential actually reaches the axon, sodium gated channels will end up opening and depolarization begins to take place. Okay, then you'll find that when depolarization has taken place, what will happen is that the action potential which is the positive charge, would travel along the axon terminal, leading to the opening of the calcium gated channels. Okay, remember, calcium is usually concentrated outside the cell than inside of the cell. One of the peculiar things that you must understand or know is that positive 30 millivolts is actually the required minimum voltage to open the calcium gated channels. Okay, so in the cell body is where the vesicles are made. Okay, so the vesicles are made in the cell body except the acetylcholine. One thing that you should take note is that acetylcholine is not a protein, though it is a neurotransmitter, and it is made in the synaptic bulb. Okay, mitochondria actually contains a lot of acetyl-CoA. Remember when I told you the formation of acetylcholine? Okay, so when acetylcholine has been formed, it is channeled into the synaptic vesicles. What will happen there is that the synaptic vesicle membrane actually has two main proteins. Two main proteins. We have what we call synaptotagmin, and we also have what we call synaptobrevin. Okay. Then there are also two proteins on the membrane of the synaptic bulb known as SNAP25 and the syntaxin. Okay. Then now the four proteins are categorized as what we call the V snares and the T snares. Now listen to this, very very important. Once the calcium ions enters, they'll activate both the V snares and the T snares. And as the result, you'll find that the synaptobrevin and the syntaxin, what they'll do is that they'll begin to intertwine. Okay, more like dragging each other or pulling each other. Okay, so similarly, that is what will happen to synaptotagmin and the SNAP25. So therefore, what we can say is that the calcium ion actually acts as a cross-link in the four proteins. As a result, the four proteins will begin to pull the synaptic vesicle towards the cell membrane of the axon terminal. Okay, and once the cell membrane fuses with the synaptic vesicle, the contents are exposed into the synaptic cleft through what we call exocytosis. Are we together? I'm seeing someone is wondering. I was just talking about neuromuscular transmission. So what happens is that the neurotransmitters will then bind to receptors on the muscle fiber, thereby generating an action potential. Okay. Now we have another part which says talks about muscle fiber depolarization. So what happens? These things you learned in class on skeletal muscle. Yes. So on step 3, we have what we call muscle fiber depolarization. Okay. So what will happen is that that action potential that was generated will tend to depolarize the muscle fiber. And when it depolarizes the muscle fiber, it will alter the electrical properties of the sarcolemma. Remember, the sarcolemma is the membrane of the muscle fiber. Okay. So what will happen is that the depolarization that has occurred will open the voltage gated sodium channels. And when this has happened, it will allow sodium ions to rush into the cell. Okay. And now, when the sodium ions have actually entered, that leads us to what we call excitation contraction coupling. I'm summarizing skeletal muscle here for you. Okay, so for some of you, you are even this is when you are learning now skeletal muscle. Okay, so it leads us to what we call excitation contraction coupling. Are we together? Before I proceed, I repeat, three. So three just has to do with the depolarization of the muscle fiber, whereby the action potential that we created gets to depolarize the muscle fiber. And when the muscle fiber has been depolarized, it will alter, or it will change the electrical potential or electrical properties of the sarcolemma. From there, since that depolarization has taken place, the sodium gated channels will open, and when they are open, they'll allow sodium to get in. Okay. Then we have what we call excitation contraction coupling. So when it comes to excitation contraction coupling, I think I'll also have to dwell a bit here. Let me expand these notes that are projected there. Okay, so what happens is that the space, okay, we have the space. So the space in which acetylcholine diffuses into in order to reach the postsynaptic membrane is what we call synaptic cleft, that we know. Okay, then one postsynaptic membrane exists. Okay, we have what we call post-junctional folds, and we have what we call ligand-gated ion channels. Specifically, we also have what we call nicotinic receptors. Now, this is the thing. When the cell is at rest in a skeletal muscle fiber, when the cell is at rest, the rest membrane potential is negative 90 millivolts. So the three things that help maintain the normal rest membrane potential, we already discussed about them. We said we have the sodium potassium multiphase pump, we have the potassium leaky channels, we have the sodium leaky channels. Okay. Now, once acetylcholine actually binds to the nicotinic receptors, the gated channels open, and what will happen is that we'll have an increased number of sodium ions entering the cell, and the potassium ions will leave the cell. Okay, so in skeletal muscle as well, the threshold is what? Negative 55 millivolts. So what you need to understand is that what you must understand is that the simultaneous influx of sodium ions and the influx of potassium ions will cause what we already talked about in the previous question. That is the end plate potential. Okay? Then the end plate potential will bring the voltage from resting membrane potential to threshold. Are we together? So by now, you must understand, or by now you know that once threshold has been reached, the activation gate on the sodium voltage gated channels will open and allow sodium to get into the cell. And once the highest voltage or level of depolarization, which is positive 30 millivolts, has been reached due to the bulky influx of sodium, you'll find that the inactivation channel closes the gated sodium channel. Okay, are we together? Remember, we are trying to expand or explain this so that you can properly understand what you're talking about. Are we together? Okay, so now the invagination on the sarcolemma. Remember, we talked about the sarcolemma, right? So the invagination on the sarcolemma is what we call the transverse tubules. Okay, transverse tubules. So this is how I'm trying to talk about the contraction, the excitation contraction coupling. I want to find where I can write. Okay, let me just write roughly here. Then we are going to try to erase. So this is the sarcolemma. Okay, then the invagination like this invagination, this invagination is what we call the transverse tubule or the T tubule. Okay, that is what I was trying to say there. Okay. Um, what question are we looking at? We are looking at question number 10, right? Yeah. So we are looking at question number 10. Yes. Okay. So now I'm still talking about the excitation contraction coupling. Hopefully, I haven't lost anyone. So what happens is that as the positive voltage is moving or traveling along the T tubule that I showed you, okay, like this. This is a sarcolemma. Then you have the T tubule like that, the sarcolemma. So as it is moving sarcolemma, then it enters into moving along the T tubule like that. What will happen is that they will meet a protein that is known as the dihydropyridine receptor. Dihydropyridine receptor. Okay. So when they meet that dihydropyridine receptor, is that they will stimulate it. So this dihydropyridine receptor is a voltage sensitive calcium channel. Okay, are we together? Are we together? I hope. So we have. This is the last question for the day. So this is actually the last question for the day. Are we together? Yeah. So from from there what will happen next is that are people still able to get me? Are you able to get me? Are we together? All right. Okay. Great. If we are still together, so we were saying that as the positive voltage will be moving along the T tubule, they will meet the dihydropyridine receptor, which is the sensitive, a voltage sensitive calcium channel, of which these things you learned during your lectures. So the sarcoplasmic reticulum actually is rich in calcium ions. Okay, is rich in calcium ions. So what will happen is that the sarcoplasmic reticulum also contains what we call ryanodine receptor. But what will happen is that when the dihydropyridine receptor gets stimulated, it gets hold of the ryanodine receptor. Okay, and when it gets hold of the ryanodine receptor, what it will do is okay. Let me just try to draw something here. So this is the sarcoplasmic reticulum. Okay, this is the sarcoplasmic reticulum. Okay, and this is the T tubule. So on the T tubule, we have what we call um, dihydropyridine receptor. So this is a dihydropyridine receptor. Okay, and then on the sarcoplasmic reticulum, we have what we call ryanodine receptor. So this is the ryanodine receptor, like that. So what will happen is that when this has been stimulated, it will get hold of this ryanodine receptor. And when it gets hold of this ryanodine receptor, it will pull the ryanodine receptor. Okay. And when it pulls the ryanodine receptor, it will lead to the opening of this sarcoplasmic reticulum. And when sarcoplasmic reticulum has been opened, you'll find that calcium ions will start flowing. And when they start flowing, they'll now go and bind to troponin. Okay. Have we followed what we've talked about? They'll now go and bind to where? To troponin, specifically troponin C part. Remember, troponin has how many parts? It has three parts, right? So they'll bind to troponin C. Are we together? And once we reach Oh, someone is asking, calcium ions come directly from ryanodine receptors. So what happens that the calcium ions are found in the sarcoplasmic reticulum. That is where they are more like stored. Okay. And then the ryanodine receptor is just a receptor which is, or we can, for lack of better terms, we can say that it's more like a door. Okay. I'm not saying that it's a door, but for you to have a clear understanding of what happens. So it's more like a door. So this ryanodine receptor, which is a door on the sarcoplasmic reticulum, closing so that calcium doesn't leak out of the sarcoplasmic reticulum. When the dihydropyridine receptor gets hold of this door, which is the ryanodine receptor, it pulls it off. And when it pulls it off, imagine that here now you have an opening. And when the opening is there, calcium starts flowing out of the sarcoplasmic reticulum. Is that clear, Muchimba? All right. Great. So once we reach positive 30 millivolts, you'll find that potassium ions will start exiting the cell until it reaches its equilibrium potential of about negative 90 millivolts. Then after that has occurred, it will lead us to what we call the sliding filament theory. Okay. Then on the sliding filament theory, what will happen, or what you need to know, what I would like to first reiterate is that the cytoplasm of the cell, of the muscle cell, is known as the sarcoplasm. Okay. So this is what will actually happen. The in the excitation contraction coupling, we saw that when calcium binds to troponin, you have a complex, tropomyosin troponin complex. Okay, so what happens is that calcium ions, just like I already mentioned, it will bind to troponin C part. Then when it binds to troponin C part, it will pull on the troponin T side. Okay. Sir, could you repeat the interaction of dihydropyridine and ryanodine receptor? Okay. So what happens is this: you have the positive voltage that has been created. I'll start from there. Positive voltage that has been created moving along the sarcoplasm, the membrane, okay, the sarcolemma rather, yeah. So the sarcolemma. So as it is moving along the sarcolemma, what will happen is that the voltage, it is coming like this. Then it reaches here where it finds this invagination known as the T tube. So the T tubule has a protein that we call dihydropyridine receptor. Okay. Then we have the sarcolemma. The sarcolemma has a protein that we call ryanodine receptor. So the ryanodine receptor is likened to a door, whereby since here we have calcium ions stored in the sarcoplasmic reticulum. So this ryanodine receptor, the interaction that occurs there is that the dihydropyridine receptor, only when activated by the positive voltage that is moving, will be stimulated now to get hold of the ryanodine receptor. And when it gets hold of the ryanodine receptor, it will pull it off. And since it is likened to a door, when it is pulled off, it will actually lead to the leaking of calcium ions. And when calcium leaves, it will go and bind to troponin C part. Is that okay? All right. Now, we are talking about the sliding filament theory. So what happens is that calcium will bind to troponin C part, and then it is going to pull on troponin T part. Hopefully you know the parts of troponin. Okay, so it is going to bind to troponin T site. So the troponin T site is the one which will then pull on the protein known as tropomyosin. Okay, so now the tropomyosin will give way for myosin. Okay, are you seeing the connection there? So it will give way to myosin, and when that occurs, myosin will then pull the thin filament. Troponin, we're talking about troponin C. What is the question exactly? I've just seen not troponin C. Okay, maybe to clear out the air, to clear out the confusion that our colleague might have. No, calcium does not bind to troponin T. I said it binds to troponin C. Okay. Remember, just for the sake of we have this. I'm trying to draw troponin. So we have that, and we can say that we also have this part, and we have this part. So what happens is that this part is for binding to uh, troponin C. Okay, and then we have this part, which you can say more like yeah, okay. Then this part is the troponin T site for binding to tropomyosin. Okay, then this part, which is troponin I site, is for binding to actin. Is that clear, Martin? Is that clear? All right, great. So where were we? Okay, so we were saying that tropomyosin will give way for myosin. Then the myosin will then pull the thin filaments, and when it pulls the thin filaments towards the middle part, trying to make the two oppositely located thin filaments slide over one another. The sliding what? Filament theory. Slide over one another towards the what? The M line. That's why I said you must know the sarcomere. Okay, so everything that I'm saying here is in line with the sarcomere. Okay, so the goal of the sliding filament theory is to see that the two actin filaments actually overlap or slide over each other. Are we together? That is what I'm trying to explain. That is here. Are we together on step 5? Then on step 6, we have now muscle contraction. Okay. So you find that the sliding of actin filaments past myosin filaments results in muscle contraction. So what happens is that the sarcomere will get to shorten, leading to muscle fiber contraction. So just to elaborate further with on this particular part, okay, what happens is that the thin filament slide over one another. We've said that. Okay. Then what happens is that the H zone, as far as you know the structure of the sarcomere, the H zone disappears. You need to know that the myosin is not moving. Okay. Myosin is not moving in this case, but it only acts as a lever. Okay. To provide leverage for the sliding part. Okay. Then you must know that titin will pull over the Z discs and they will get closer to each other. Then the thick filament doesn't move as well. The A band stays the same. Someone is confused. Yes, myosin ain't moving. What it does, it only provides, acts as a lever. Okay, or if, for lack of better terms, you can say it acts as a simple machine. Okay, are we together, Mchimba? All right, great. So after that has occurred, the I band decreases in size. Then we are done with the muscle contraction. I think I've helped you remember skeletal muscle contraction in detail. Okay, so that has to do with the stroking and the like, but in this case, in your case, it is not that much relevant. Okay, but if you want it to be talked about further, you can reach out to me for further elucidation. Are we together? I think we should end here. Is there any other question? Let me see. Okay, let's do one last question. Then we are done for the essay questions. Um, let's talk about question 22. Then we'll await for the next session with that you people are going to have with my colleague. Question 22. So you've seen question 21 or question 10 was talking about contraction. Then question 22 is talking about relaxation. Okay? It's talking about relaxation. So the question says, explain the process of skeletal muscle relaxation in humans. Skeletal muscle relaxation in humans. Okay? Now, this is interesting, right? I'm sure most of you are learning and the like. So, when it comes to relaxation of skeletal muscle, you need to understand that the relaxing of skeletal muscle fibers and ultimately the skeletal muscle begins with the motor neuron, which stops releasing its chemical signal, that is acetylcholine, into the synapse at the neuromuscular junction. Okay, so what happens is that the muscle fiber will depolarize. So when it depolarizes, it closes the gates in the sarcoplasmic reticulum where calcium was being released. Okay, then the ATP driven pumps will now move calcium ions out of the sarcoplasm back into the sarcoplasmic reticulum. Okay, then this the above occurrence, which is the taking back of calcium into the sarcoplasmic reticulum, results in the reshielding of the actin binding sites on the thin filaments. Okay, now let me try to expand what I've just said in those few points. You must understand that normally, let me take you back again. Normally, the peak potential we say that is positive 30 millivolts. So that positive 30 millivolts activates the inactivation gates on the voltage gated potassium channels. Okay, so what will happen is that during the peak potential, which is positive 30 millivolts, calcium ions tend to leave troponin C part. Okay, they will leave the troponin C part. And what will happen is that the troponin T part will then leave the tropomyosin in return, and the active sites on actin will be blocked again. Okay, so what will happen is that the calcium ions will actually have to go back to the sarcoplasmic reticulum, and two channels are present on the sarcoplasmic reticulum. Okay, but remember, calcium is more concentrated in the sarcoplasmic reticulum because why is it concentrated, or why is it being held tight, so tight that it can't leave, is because of a special protein known as calciquestrin. Calciquestrin. Okay, so calciquestrin is the one that normally pulls them so strongly held together in the sarcoplasmic reticulum. Okay, so for calciquestrin, there are two main channels through which calcium gets back into the sarcoplasmic reticulum. Okay, we have, remember, it is moving against the concentration gradient, that is active transport. Okay, so we have what we call the sodium calcium exchanger. Okay, sodium calcium exchanger, where calcium goes into the sarcoplasm sarcoplasmic reticulum, and then calcium goes in. Then we also have the calcium ATPase pump. Calcium ATPase pump. So after calcium has been taken back to the sarcoplasmic reticulum, the thin filaments and the Z disc will go back to their normal positions. Then, by so doing, we have relaxation. Calsequestrin is a special protein that gets to pull the calcium ions so strongly in the sarcoplasmic reticulum. Having said that, having said that, have we revised when it comes to the essays and the short notes?